Hello students, I am Dr. S. Gomathi, Assistant Professor, belongs to the Department of Biochemistry, School of Allied Health Sciences, VMRF-DU, Salem. Today we will see about amino acid metabolism in human health. Before entering into the topic, we will see about some basics of amino acids. So we all know about amino acids are the building blocks of protein synthesis. The basic structure of amino acid contains central carbon atom and it contains two functional groups like carboxylic acid group and amino group, and it has a R groups. So it contains one carboxylic acid group and amino group. So these functional groups has a several beneficial effect. They are structural elements and energy sources of cells necessary for normal cell growth, differentiation and function. So amino acid metabolism disorders have been linked with a number of pathological conditions including metabolic diseases, cardiovascular diseases, immune diseases and cancer. So we will see about some functions of amino acids. Besides serving as building blocks of proteins, amino acids have multiple regulatory functions in cells. These nutrients are circular for growth, development and health of animals and humans. So these have several functions including break down food, grow and repair body tissue, make hormones and brain chemicals like epinephrine and norepinephrine, and it provides an energy and it sustains a normal digestive system. Because digestive system contains several enzymes, it involves in the digestion of macromolecules, and it boosts the immune system acting as immunoglobulins, and it builds the muscle, and it maintains healthy skin, hair and nails because healthy skin contains the collagen, it is a protein, and hair, skin, nails also contains the protein like keratin protein. So same as I said before, these are the different functions of amino acids. Now let's move on the topic metabolism. All we know about metabolism refers to the complex network of chemical reactions that occur within living organisms to maintain life. These reactions are responsible for converting nutrients into energy and building blocks for growth, repair and maintenance of cellular structure. So these metabolism broadly classified into two types, so anabolism and catabolism. In anabolism, the simple molecules are converted into macromolecules, and in catabolism, from simple molecules the energy will be produced. Before enter into the metabolism of amino acids, we should know about amino acid pool. So what is amino acid pool? It is a grand mixture of amino acids available in the cell derived from dietary sources or degradation of protein. Since proteins and amino acids are not stored in the body, there is a constant turnover of protein. Unlike carbohydrates and fats, amino acid does not have a storage form. So it should contain the amino acid pool for anabolic and catabolic processes. Because all proteins in the body are continuously degraded and newly synthesized, not stored in the body. So amino acid pool are present in our body for the metabolic processes. For these amino acids, the sources of amino acids are from food dietary sources, tissue proteins and non-essential amino acid synthesis from body. It makes the amino acid pool. So this amino acid pool is used for new body proteins, specialized products, synthesis of specialized products like amines, porphyrins, nucleic acid bases and catabolic processes, that is energy gaining process. So this pictorial representation shows the utilization of amino acid pool as well as the sources of amino acid pool. So amino acids are derived from dietary sources, tissue protein and non-essential amino acid synthesized from the body. These make the amino acid pool, and from these amino acid pool anabolic and catabolic reactions are carried out. In the anabolic reaction, proteins will be synthesized and other nitrogenous compounds also synthesized like amino sugars, nitrogenous bases, purines, pyrimidines, neurotransmitters, niacin, creatine and heme. In the catabolic process, the amino acids are converted into keto acid and ammonia. These keto acids will be utilized for the synthesis of glucose, ketone bodies and also in the energy production. The ammonia will be converted to urea and it will be excreted through urine. So the maintenance of amino acid pool is very important for the protein turnover. So let's see about the catabolism of amino acids. So catabolism of amino acids is nothing but breakdown of excess amino acid. It takes place primarily in the liver. So it takes place in the two steps. First one is removal of amino group to form ammonia, which later is disposed after conversion into urea. The second thing is breakdown of the remaining carbon skeletal which remains after removal of amino group. So this is the overview of the amino acid catabolism. The intracellular proteins are pooled into amino acid pool. So the excess amino acids first undergo the transfer of amino group and the remaining carbon skeleton will enter into the TCA cycle. So the amino group will be utilized for the other nucleotide synthesis and non-protein compound synthesis, as well as it is enter into the urea cycle and it excreted in the urine. The keto acids again enter into the TCA cycle as intermediates, and also the keto acids are utilized for the glucose production in the process of gluconeogenesis and also energy production. So the catabolism of amino acid takes place in the 3 steps. First one is transamination, so in which amino group is transferred to the keto group, keto acid, and second step is deamination. In this, the amino group will be removed and ammonia will be formed. This ammonia will be enter into the urea cycle and urea will be formed and it is excreted in the urine. So this picture represents the fate of the amino group. Initially amino acid is converted to glutamate in the transamination reaction. After that glutamate is converted, from glutamate ammonia will be removed, then this ammonia is enter into the urea cycle. So the fate of amino group is involved in the 3 steps: transamination, deamination and urea cycle. In transamination reaction, amino group will be transferred to the keto acid, forms the glutamate, and in deamination process amino group will be removed, and in urea cycle the ammonia enter into the urea cycle, converted to urea and excreted in the urine. So this is the transamination reaction. So amino acid combined with the keto acid to forms the amino acid glutamate and keto acid in the presence of amino transferase. So in the transamination reaction, the amino group is transferred from one amino acid to a keto acid, forming a new amino acid and a new keto acid. This reaction is catalyzed by the amino transferases. The second one is deamination. This is the removal of amino group. From the removal of amino group, form an amino acid resulting in the formation of ammonia and a keto acid. This can occur via the oxidative deamination or non-oxidative deamination. So in the oxidative deamination, when an amine group is removed from a molecule by the process of oxidation, the reaction is called an oxidative deamination reaction. These type of reactions largely occur in liver and kidney. These reactions lead to the production of alpha keto acids, etc. via the amine groups. Then we will see about non-oxidative deamination. In this, the removal of amine groups from a molecule via reactions, all except the oxidation reaction is called non-oxidative deamination reaction. The main types of reactions that are involved in this process are reduction, hydrolysis and intermolecular reactions. So this reaction shows the non-oxidative deamination reaction. Histidine converted to the urocanic acid by the histidase enzyme by removal of ammonia. Same as L-cysteine is converted to the amino acids. Initially the intermediate is formed, then it is converted into pyruvic acid. During this process ammonia will be removed. Same as serine is converted to initially amino acid, then it is converted to pyruvic acid. During this process ammonia is liberated. So this ammonia is entered into the urea cycle. In urea cycle ammonia is converted to carbamyl phosphate, then carbamyl phosphate and ornithine it is converted to citrulline. Finally urea will be liberated. The process by which excess nitrogen is converted to urea in the liver and excreted in the urine. Let's see about the fate of carbon skeletons. So still now we have seen about the fate of ammonia. Hereafter we will see about the fate of carbon skeletons. The carbon skeletons derived from amino acid degradation can enter into several metabolic pathways based on their structure and cellular requirements. So all 7 products will be act as a intermediates in the TCA cycle, as well as it will be used for the ketogenic product synthesis and also glucose production. These are pyruvate, acetyl-CoA, acetoacetyl-CoA, alpha-ketoglutarate, succinyl-CoA, fumarate and oxaloacetate. All these products enter into the citric acid cycle and are either involved in the gluconeogenesis or in ketogenesis, or either oxidized to CO2 and H2O with the production of energy. So here some of the amino acids acting as a glucogenic amino acids and some of the amino acids acting as a ketogenic amino acids and some amino acid acting as both glucogenesis as well as ketogenesis. So glucogenic amino acids are converted to pyruvate, alpha ketoglutarate, succinyl CoA, fumarate or oxaloacetate. These are the glucogenic amino acids, example alanine, glycine, serine. So ketogenic amino acids are converted to acetyl CoA, acetoacetyl CoA. The examples are leucine and lysine are ketogenic amino acids. So amino acids which involves both glucogenesis and ketogenesis are isoleucine, phenylalanine, tryptophan and tyrosine. Gluconeogenesis is nothing but some amino acid carbon skeletons can be converted into intermediates of gluconeogenesis. The synthesis of glucose from non-carbohydrate precursors called as gluconeogenesis. So this process is especially important during the fasting or prolonged exercise when glucose levels need to be maintained. And ketogenesis are nothing but the amino acids raised to the formation of ketogenic product like acetyl CoA or acetoacetyl CoA. These are the precursors for the synthesis of ketone bodies. Ketogenesis occurs predominantly in the liver, provides an alternative fuel source for the tissues, particularly the brain during periods of prolonged fasting or low carbohydrate intake. And energy production. Amino acid carbon skeletons can also enter the citric acid cycle as intermediates where they undergo oxidation to produce ATP through oxidative phosphorylation. This process contributes to the overall energy balance of the organism and is essential for cellular functions. So this picture shows the overview of amino acid degradation. Some of the intermediates are entered into the TCA cycle and some of the amino acids are used for the precursor of gluconeogenesis, glucose production. Amino acids also involved in the synthesis of other specialized products like heme, purine, pyridines, hormones, neurotransmitters and biologically active peptides. Histamines plays a central role in many allergic reactions. This is also derived from the amino acid. Also neurotransmitters derived from amino acids include gamma aminobutyrate, 5-hydroxytryptamine, that is serotonin, dopamine, norepinephrine and epinephrine. These are the derived from the amino acids acting as the neurotransmitters. So this pathway shows the tryptophan neurotransmitters. So here tryptophan serves as the precursor for the synthesis of serotonin. So this pathway shows the creatinine biosynthesis. So creatine compound is a high energy producing compound. So this is also derived from the amino acid. So creatine synthesis occurs predominantly in the kidneys and the liver. The synthesis of creatine begins in the kidneys using the amino acids arginine and glycine. The formation of guanidinoacetate from these two amino acids is catalyzed by the enzyme glycine aminotransferase, also called L-arginine glycine aminotransferase. So let's see about the disorders of amino acid metabolism. During the metabolic process, some of the enzyme is deficient, so the breakdown of amino acids will be slow. So the inherited disorders will be formed. These are the amino acid metabolic disorders like that: phenylketonuria, biopterin defects causing hyperphenylalaninemia, hyperphenylalanemia due to DNA JC12 deficiency, tyrosinemia type 1, type 2, type 3 and alkaptonuria. Phenylketonuria is formed due to the phenylalanine hydroxylase deficiency enzyme. Same thing, tyrosinemia type 1, type 2, type 3. These are also formed due to the deficiency of fumarate acetoacetate hydrolase enzyme, hepatic stolic tyrosine aminotransferase enzyme, 4-hydroxyphenyl pyruvate dioxygenase enzyme, and alkaptonuria is formed due to the deficiency of homogentisate dioxygenase enzyme. And maple syrup urine disease is formed due to the deficiency in the branched chain amino acid metabolism pathway. Here branched chain alpha ketoacid dehydrogenase complex is a deficient enzyme. Due to this enzyme, maple syrup urine disease is formed. So due to this, brain will be affected. Same as cystinuria, lysinuric protein intolerance, Hartnup disease. These three are formed due to the defect in the transport mechanism. And hyperlysinemia type 1, it is formed due to the deficiency of alpha aminoadipic semialdehyde synthase enzyme. Thank you for watching my class.